As we know, we're all gathered here together to enhance the student experience through philanthropy and engagement from all of us who are proud to send our children to Tufts every single fall. As we experienced the faculty, this incredible musical and performance groups over the weekend, and the talented student athletes throughout this weekend, we remember what inspires us to support Tufts' mission with our philanthropy. Lauren and I are proud parents of two jumbos, one who graduated last year in the Arts and Sciences program, and a current sophomore who is in the engineering program, Alex. Before we get started, maybe just two quick housekeeping notes, if I may, before we begin. We'd ask you that you please silence your cell phones and take note of the exits, please, in front of you and in back of you. And now, Lauren and I have the pleasure to introduce Tony Monaco, Tufts' 13th president. An accomplished leader, scientist, and teacher, Dr. Monaco brings to the Tufts presidency deep-rooted commitments to academic excellence, diversity, access and inclusion, a global perspective, and a keen awareness of the power of higher education to impact individuals and society. He is leading Tufts in advancing its long-standing commitments to innovation, collaboration, civic life, global perspective, and transformative experiences for our students. In recognition of his outstanding leadership in education and science, Dr. Monaco was elected to the American Academy of Arts and Sciences in 2018. Founded in 1780, the Academy is one of the nation's most prestigious honorary societies. It brings together exceptional scholars, leaders, artists, and innovators to share knowledge and address challenges facing the world today. Please welcome Tony Monaco.
Well, thank you, Lauren and Peter, for that warm introduction, and welcome to all of you for this wonderful weekend where you'll have the chance to experience what life is like on campus as your children do. I hope yesterday you had the chance to meet some of our leaders, our new provost, Nadine Aubrey, who joins us from Northeastern University, as well as some of the deans, Glazer, Q, Bauer, and Solomon of our various schools, and hopefully they shared some of their vision for Tufts. Also, you had a chance to meet some of our advising or residential life staff and learning, career staff, as they outlined and explored some of the many resources that are available to your students here at Tufts. Today, we really want you to spend some time learning, like your students do, from some of our amazing faculty. One of the best parts of Tufts is that these individuals are at the cutting edge of their field, and they really do bring the excitement of that into the classroom, challenge our students, and ask them to join them on their teams, in their scholarship, to also contribute to these fields. Some of the notable faculty you'll hear from is Laura Gee, Associate Professor from the Department of Economics, discussing behavioral economics, understanding irrational behavior, and using it to make better decisions. Frank Lehman, Associate Professor from the Department of Music, sharing his work in film scoring with a class titled Must Go Faster, Music in the Contemporary Action Scene. Helen Su, Professor from the Department of Civil and Environmental Engineering, discussing her research into how air pollution impacts aging and health outcomes. Keith Maddox, an Associate Professor, and Sam Summers, Professor of Psychology, discussing the science of stereotyping and implicit bias. And Floor Vandervelde, Professor of the Practice at the School of the Museum of Fine Arts at Tufts, who will share a class on why art is essential for 21st century learning. But as many of you know, when you contact your students, they're not spending all of their time in the classroom. Many of them will be enjoying all the opportunities available through clubs, athletics, music performances, dance, and other activities. And this weekend, we'll have a lot of that on display. I hope you'll enjoy some of our athletic games, including football, men and women's soccer, field hockey, and also the Avenue of the Arts performance, where we'll see such amazing talents from our students. Let me share with you some brief highlights of this year. Matriculation started with a great ceremony where we could welcome the class of 2023. What an accomplished group. And we're so proud that our engineering school is now 51% women. We also had a little bit of a rain cloud burst during that matriculation. For those of you who were here, I hope you were impressed on how quickly we could get the ponchos out. It was quite a sight to see from the stage. We've also had some outstanding speakers, and I think this is truly one of the opportunities that our students get because of the closeness to the Fletcher School and also the activities in the Tisch College around voting, participation, and democracy. We have some incredible speakers, many in the political arena, who come through and share their insights and their differing views with your students. We had Supreme Court Justice Sonia Sotomayor, politicians Beto O'Rourke. We had anti-Semitism expert Deborah Lipstadt, and other folks like Carl Rove and Ed Markey. One of the best celebrations we had this year was the Africana Center's 50th anniversary of their center here on campus. We had more than 200 alumni come back from various decades, going right back to the 60s. It was quite a celebration. As many of you know, Tufts' mission is as a student-centered research university. We put the students at the heart of what we do, but of course we have so much scholarship and research that has such an impact on society. This morning's sessions will give you a sense of faculty research, but we also do a lot of work here to support student life. One very important issue, which has increasingly and almost alarmingly overtaken much of the support we give, is around mental health. It's an important topic here at Tufts as well as elsewhere in higher education. We're fortunate to have excellent on-site resources in place, particularly here on the Medford campus and for our SMFA students. Not only our mental health counseling service, but we have a network of surveillance and support from our Dean of Student Affairs and Residential Life. I think they are very good at breathing in chaos and breathing out calm. But they face challenges nonetheless. The utilization of resources is now at a very high level. 28% of our students, both at the undergraduate level and also pro rata into our graduate and professional students, are availing themselves of these services. Over the last 15 years, that's a 1% per year increase. So you can imagine that a lot of the alarm at the moment is because we're creaking at the seams on how we're dealing with such volume. If that wasn't the only problem, we're also seeing an increase in the number of acute cases, students who present with distressing symptoms and panic attacks. Many of them are also seeing this as the first episode, and this reminds you, this is the age group where major depression and bipolar disorder and other major mood disorders have their onset. Because of these factors, 2.5 years ago, I decided to co-chair with Paul Summergrad, the chair of psychiatry at Tufts Medical Center, a task force focused on this mental health crisis. We took a very deep dive into services and resources, how we could think about prevention as well as increased support for those that were experiencing problems. We took a hard look at our policies, particularly how we had a surveillance network to pick up students who were failing in academics, which of course is one of the first signs that someone may be having trouble. When we do surveys, 75% of our students say that their academic success is impeded because of mental health or emotional issues. So we think it's something we need to take seriously. We've had a very positive response from putting our report out. I urge you to read it. With many recommendations that we were already able to implement because of wonderful collaboration with our faculty and staff, including many that are involved in this surveillance network. Now that the task force has completed its work, I will be happy to co-chair or chair a steering committee which will advance the implementation and the looking at outcomes based on our work. We are also joining forces with other colleges and universities nationwide to tackle this problem. We have joined closely with the JED Foundation, which is known nationwide as a leader in mental health prevalence and treatment. We also recently opened up an office and support at 200 Boston Ave, just down the street, with The Haven, which is a private organization that was very successful on the West Coast around substance abuse, which is very much related to some of the mental health issues that our students are currently experiencing. The Haven provides treatment, support, and a lot of activities for students who are coming back or seeking support for substance abuse and related mental health issues. I know that these are issues that many of you have experienced in your own homes or through relations. I know that it's something that we're all struggling with. I will have a session at 11:15 in the Granoff Music Center in the Fisher Performance Room to discuss and take your questions about these issues. I'll be joined by Julie Ross, the director of our Counseling and Mental Health Service, as well as Kirsten Belling, our Associate Dean of Student Accessibility and Academic Resources. As many of you know, it's not just getting help and treatment and support from the counseling service, but it's also understanding how to navigate all the educational accommodations that many of our students need to help them with these issues. I really look forward to talking to you as parents about these issues and taking your questions. We've also invested a lot in other aspects of student life, and that is the built environment in which they live and do their work. We've completed a major renovation of Miller Houston. I hope you'll have a chance to see how we've opened up those dorms, made them disabled accessible, as well as offered new opportunities for juniors and seniors in residences in wood frame houses in Medford, in which we're calling community housing. Barnum Dana, the building right on the quad that used to house biology, is now the home of Tisch College, Film and Media Studies, Studio Arts, and Tufts Institute for the Environment. So I hope you'll have a chance to look at that renovation. If you go down to College in Boston Ave, you'll see that we've started work on the Joyce Cummings Center, a building which will house our ever-growing programs in computer science, data science, economics, and mathematics, as well as a range of new teaching spaces. We hope that it'll open up in 2021 at the same time as the Green Line extension coming from Lechmere, 4 stops up to directly to our campus. We also are adding a squash facility at the back of Gancher. We hope to open this up in the spring so our squash team will be able to practice on site. But of course, like all of our fitness facilities, it'll be opened up to students, faculty, and staff. As many of you know, we're constantly trying to raise funds to support our mission, and we have a Brighter World campaign, which we are in the last couple years of. Quite a ambitious goal of $1.5 billion, but supporting transformational experiences for our students, supporting faculty research, helping to retain the best faculty here at Tufts for their teaching, for their innovation, and for the spaces they need to carry out their work at the standards that we hope that they will achieve. But we also think about how we affect our local communities, our global communities. What impact does having students come through here? One of the best things I get to see as president is how over 4 years they develop not only intellectually, but also personally and developmentally. So when they leave this place, they really do understand their position as global citizens. Wherever they may end up, they will take that spirit and invest not only in their careers and in their work, but also in their local communities and in their democracies. We've done very well raising money. We're on target for this goal, and thank you to all the parents that have shown their support. We've raised $200 million just for financial aid to support making Tufts as diverse as we can. But now it's my pleasure to introduce to you one of the most exciting faculty that I've gotten to know during my 9 years here at Tufts. That's Michael Levin, a professor in our biology department who holds the Vannevar Bush Endowed Chair. He also currently serves as director of Tufts Center for Regenerative and Developmental Biology, as well as the Allen Discovery Center. Mike is a jumbo himself, one of the best of our undergraduate alumni, where he got his degrees, Bachelor of Science in Biology, as well as Computer Science. So a visionary, even at a young age, when he saw how these two disciplines were going to come together to really understand some of the complex issues facing biology. He got his PhD from Harvard University for the first characterization of the molecular genetic mechanism undergoing how our bodies form left-right asymmetric patterns. How does the heart know to be on one side and not the other? How does the liver know to be on one side and not the other? That's thanks to a lot of the work that Mike did as a PhD student. This work is on Nature's list of the 100 milestones of developmental biology of the century. He's recently received honors as Scientist of Vision Award and a Distinguished Scholar Award. Mike, in 2016, was one of the first 2 scholars to receive a grant from Microsoft co-founder Paul Allen to create the Allen Discovery Center at Tufts. This is a $10 million grant, and it's intended to fund research on the frontier of life sciences. And I believe you'll see from Mike's presentation just how exciting his work is for the future of biology. Thank you very much, and I hope you enjoy the weekend.
Good morning. It's a great pleasure to share with you today some of the profound mysteries that keep our group up at night. And if anyone is interested in getting in touch with us or you have more questions afterwards, please feel free to contact me through these websites. We can start off by thinking about the fact that if we could control anatomy, that is, have complete and full control of the complex three-dimensional structure of living form, basically, almost every issue, with potentially the exception of infectious disease, every other issue of biomedicine would be solved. If you could control three-dimensional structure of living things, we could repair birth defects, we could cause the regeneration of organs after traumatic injury, we could reprogram cancer, we could reverse aging and degenerative disease. And in fact, if we understood how cells compute, make decisions to get together to build complex organs and structures, we could also make synthetic living machines. And in fact, this would spill over from biology into making robust novel computing devices and even new artificial intelligence platforms. So the understanding of shape and of biological structure is really fundamental for unlocking the promises of regenerative medicine in this post-genomic era. We all know now that we can sequence genomes, and I'm here to talk to you about some of the next steps. You know, once we have these genomes, what happens next? And in particular, I want to talk about some of the fundamental problems, and then through the eyes of the students, the undergraduates that in our group have made really important advances in these areas, tell you some of the things that we're thinking about. And so this is a view. This is sort of a front entrance to our lab. This is our Allen Discovery Center is housed in this new science and engineering complex. And my group works at the intersection of three areas, so biophysics, computer science, and cognitive science. And this all comes together through the hard work of not only the PhD students, staff scientists, and so on, but really the undergrads. And the undergraduate students have made an amazing set of advances here that I would like to share with you. So let's warm up by thinking about plasticity. Now, this is a caterpillar, which is a soft-bodied creature that has to crawl around and chew leaves. And this has to turn into a stiff-bodied creature that flies and drinks nectar. Have completely different behavioral repertoires. So during this process of metamorphosis, not only is the body completely reshaped, but in fact the brain is basically liquefied, it's taken apart, and reassembled back into a new kind of architecture that is more appropriate to drive this sort of creature. But the most amazing part, so that's amazing already, but the most incredible part of this is that memories that are formed in this caterpillar brain are retained in the butterfly, even though the brain is taken apart and basically recreated from scratch. So this nexus of the plasticity of the information in the brain, but also the three-dimensional structure of both the brain and the body, really brings to the forefront some profound questions that we don't know the answers to. And in fact, and this is some work from our lab, in these planaria, these flatworms, which I'll tell you more about in a minute, one of the amazing things about them is that they regenerate every part of the body. So if you were to cut off their head, this tail piece right here would regrow a brand new brain from scratch, basically. And what we found is that memories that this animal generates during its lifespan, if you cut off the brain and let the tail regenerate a brand new head, this now keeps the original information. And so this really raises some profound questions about what is a memory? What does it mean to store a learned piece of information in biological tissue? In fact, when that tissue can be either rearranged or restored from scratch. And not only is this important for deep questions of philosophy of mind, but really also for the biomedicine of brain disorders, because very soon we're going to have adult patients with, let's say, 5 decades, 6 decades of experience having stem cells introduced into their brain to treat degenerative brain disease. What happens to the memories, the personalities of these patients? And how can information persist through this kind of remodeling? So what's really important also is that this kind of plasticity is not only for the brain, it in fact also exists in the body. And so we all start life here as one cell, the fertilized egg, and this thing reliably self-assembles into one of these many remarkable morphologies. And so we need to understand where this pattern comes from. And in fact, this is similar to a talk I give to middle school kids, where I start off showing an egg and saying, well, out of this egg could come lots of different types of morphologies. What actually determines what comes out? And even 7th and 8th graders immediately will say, well, it's the DNA, it's the genome. But what does that actually mean? This is not a simple question, and let's talk about this for a second. Individual cells are highly competent. This is a single cell creature called the lacrimaria. And you see there's no brain here, there's no nervous system, there's really only one cell, in fact. And you see what it's doing. It has remarkable control over its anatomy. It's going around and picking off bacteria that are in its environment. It's feeding, and it has incredible real-time control over its shape, its physiology, and its behavior. And so if single cell, and we all used to be single cells, so, yeah, so millions and millions of years ago, before multicellular creatures evolved, this is what was around. And the question is, if these individual cells are very dynamically competent to handle their own needs, how and why did larger creatures evolve? And here you can see that in bodies, in embryos, so this is a frog embryo, this is a zebrafish embryo, all of these cells cooperate with each other to build very large structures, such as whole entire bodies. And so the fact is that when cells transition from being free-living life forms to making up a body, they didn't lose all these intelligent capacities that they had, but they harnessed them in a different purpose. So instead of just handling the business of a single cell, they now cooperate to make much larger structures. And the boundary or the surface of the self, the edge of the coherent organism has grown drastically through evolution. And so we need to understand, we start off life as a collection of embryonic blastomeres, and we sort of expect that reliably this is a cross-section through a human torso. You can see the amazing complexity, all the different tissues that are arranged in precisely the right way, and all normal babies eventually end up like this. So we need to understand, where does this anatomical arrangement comes from? Because if you look at the genome, if you actually read what's in the genome, this is nowhere to be found. What's in the genome are sequences of specific proteins that are inside every cell, but we cannot look at a genome and read out the three-dimensional structure of what's going to come out. So it's not directly in there. So the question is, where does this shape come from? How do the individual cells know what to build and when to stop? And how far can we push this? As engineers or bioengineers, we want to understand how would we convince these cells to build something different, something new, and of course how to repair it if there's damage. So all of these are profound questions that are still with us. Now, the important thing about embryonic development is that even though it's highly reliable, it is not hardwired. So for example, if an early mammalian embryo is split in half, you don't get 2 half embryos, which is what you would get if you split any of our artifacts, a computer or anything else like this. You split it in half, but an embryo is incredibly dynamic in that each half will notice what's missing, recreate the missing part, and then you get 2 perfectly normal monozygotic twins. You can do the opposite, which is to take 2 mouse embryos, mush them together sort of like a snowball without any regard for where the different cells are going, and you will get 1 perfectly normal animal out the other end. So that already tells you that this process has a kind of intelligence to it. It is not a hardwired set of steps. It makes up for these kind of radical deformations. And in fact, this is not only embryonic. So some animals, such as this salamander known as the axolotl, this guy regenerates its limbs, its jaws, its eyes, its ovaries, its spinal cord, portions of the heart, and portions of the brain during adulthood. So if you were to amputate, and in fact, they do this naturally, they sort of bite off each other's legs all the time in the wild, and so it's not just an experimental thing. So if this happens, they will rapidly regrow a new leg, and the new leg is indistinguishable from the original, okay? And the most amazing part, lots of people are working on the regenerative medicine question of how do we kickstart this in humans, okay? That's obvious. But one of the most remarkable aspects of this is that it knows when to stop. Think about this. This is incredibly rapid cell growth, but instead of continuing to grow out of control, as you might in a tumor, it knows exactly when a correct axolotl limb has formed, and then it stops. How does it know what a correct axolotl limb looks like? How does it know when to stop? So these are the kinds of things that we work on. And in particular, this animal, these Planaria, they are champions of regeneration. They're complex. They are our direct ancestors. They have a true brain. They have bilateral symmetry. They're not like an earthworm or something like that. They have most of the same neurotransmitters that you and I have. And they have some amazing properties. First of all, you can cut them in as many pieces as you want. The record is 275. And every piece will grow exactly what's needed, no more, no less. So every piece knows what's missing and will regrow what's needed. In fact, what this has led to is that these creatures are effectively immortal. There is no such thing as an old Planarian. The individual cells senesce and die and fall off, but the animal regenerates them constantly. And so the animals that we have here in our lab are in direct physical continuity with animals that were here almost 500 million years ago. These are the same animals. So if the theories of aging that says that a complex animal like this has to die, it has to have a limited lifespan because thermodynamics, you accumulate errors, things like this. I think these animals are telling us that's wrong. Immortality with a complex body plan and learning, and in fact learning capacity and memory, is possible. These animals are doing it. So I'm very interested to understand how this works. But I should say that regenerative ability is not only for these so-called lower animals. So the human liver, of course, is highly regenerative, and this was known as far back as the classic Greeks. I don't know how they knew this, but they did. Deer will regenerate up to about 1 meter of new bone every year, so the antlers fall off. And then at a rate of about 1.5 centimeters per day of new bone growth while this is growing back. Here's an adult mammal, a large adult mammal, that's growing new bone at the rate of 1.5 centimeters. And in fact, even kids, human children below the age of somewhere between 7 and 11 are able to regenerate their fingertips. Lots of people don't know this, that actually if a small child loses a fingertip, if you don't sew it over, it will actually regenerate. And then we lose this ability with age. So it's some degree of regenerative capacity still exists. And understanding this process is not only vital for medicine and for regenerative medicine, but also for robotics and robust engineered systems. So for example, we make these highly complex, very expensive sorts of autonomous systems. You might send them to Mars or somewhere else, and if there's some sort of damage, whereas this biological system will heal itself nicely, this is pretty much it. And so for the robot, and so we need to understand there are lots of our collaborators are working in soft body robotics and autonomous systems to try and learn from the biological case to see what would we have to do, what would we have to do to enable these things to repair themselves. Okay, so I want to give you really a good example of this goal-directed behavior that living systems are able to do. So this is something we discovered a few years ago. It's known that a tadpole, in order to become a frog, has to rearrange its face. So during the process of morphogenesis, the metamorphosis, rather, the eyes have to move forward, the nostrils have to move out, the jaws, everything sort of rearranges. And it was thought that this was a rote process, meaning that every tadpole looks the same, every normal frog looks the same, so if every organ always moved the same amount in the same directions, everything would be okay. So what we did was we looked at what happens to what we call Picasso frogs, and these are frogs where the tadpoles start off in very, very abnormal positions. So the eyes are back here, the jaws are over here, everything's sort of mixed up. And what happens remarkably is that during this process, they basically turn into pretty normal frogs. So what the frog genome does not encode is a set of specific movements for the different organs. What it does encode is a flexible system that keeps moving and keeps rearranging the different organs until they get to the correct state. So this is a kind of feedback loop. It's a homeostatic cycle where it knows what the correct set point is, and it will continue to move to minimize that error. It tries to minimize the difference between where we are now and where we need to be. And so this raises a number of important questions, which is how do the cells make group decisions, and how do they recognize what pattern is correct or incorrect? But it highlights this really remarkable ability of cells. When they get together, they can work towards specific endpoints. They represent a specific stop state, and they work to reduce error, and they cooperate, striving to achieve this particular state, despite various deformations that you might make. And so what happened then during evolution is that by getting together, by forming networks, communicating networks of cells, they went from very tiny cells that basically just managed the conditions right around a single cell surface into managing a much bigger thing. They now, the self has now expanded so that they can now pursue very large goals compared to themselves. So the goal of maintaining a whole limb or a whole body or a whole face. And so we study these computational processes whereby individual cells merge into networks, into tissues, organs, organisms, and perhaps even colonies. You can think about ant colonies as a kind of organism as well. Maybe even human societies as a kind of organism. But with each new step in organization, the goals, both in duration of time and the scale of how far they propagate from the creature, get bigger and bigger, so that they're now able to work towards these very large kinds of goals. And in fact, this is very important biomedically, because one of the things that was learned, and this was not our work, this was discovered decades ago, that you could actually take cancer cells and introduce them either into a regenerative or into an embryonic environment, and they become normalized. The cues from the surrounding environment that tells every cell what to do, in this case to rebuild the arm of the salamander, those cues can take a cancer cell, which has basically sort of defected from the body plan. It's gone off on its own, kind of reverted to this amoeba-like ancient existence. This can be overridden. And so if we had a better understanding of how these cues control cell direction in terms of the purpose that they're trying to implement, we could have a much better way to address cancer than, let's say, toxic chemotherapy. So this kind of reprogramming is a profound thing to try to understand. So it's important to think about what is needed for this kind of transformative revolution in biology. And one revolution that we can think about is what happened in computer science. Computer science gave us this amazing information technology revolution because the field understood that what's really critical is to control information, not just the energy or matter, and they focus on the algorithms, not only the hardware. And what I mean by this is what computer programming looked like in the 40s. So if you wanted to program a computer, you literally had to interact with the hardware. You had to go and rewire the thing manually. So you had to work at this level. You can imagine that doing anything complex at this level is really hard. But over time, we've developed things like assembly language and then really high-level languages, and we don't touch the hardware. So when you, on your computer, when you switch from Microsoft Word to Adobe Photoshop, you don't have to get out your soldering iron and start rewiring, right? And the reason is because you've got really good hardware. It's because the hardware is able to change what it does based on inputs, signals, experiences that you give it with your keyboard, not by rewiring the hardware. So biology currently is very much focused at the level of hardware. Some of the most exciting advances in the biosciences are single molecule advances, genome editing, single molecule precision techniques. And these are extremely important, but they're just the tip of the iceberg because this is still at the level of hardware. We're still rewiring. What we really need to understand is to move beyond this machine code and to figure out how to control the software that orchestrates the cells towards specific endpoints, such as rebuilding complex structures. So in order to do this, so this is what my group tries to do. And in order to do this, we have, of course, the various scientists at all levels, from senior staff scientists to undergraduates, and the model system. So we, of course, use these planaria. We use frogs, which make tadpoles and eggs and frog embryos. And then we even use these slime molds. So this is kind of an amazing aneural organism that's able to make decisions about what's around in its environment and explore. It likes to explore larger objects versus smaller ones. So these are all things that we do. And what we've basically found over the last, let's say, 2 decades is that just like in the brain. So in the brain, you have, this is well understood, you have some hardware, which are basically electrical cells, electrically active neurons, and they talk to each other through these little connections known as electrical synapses. And this hardware underlies the software of the brain, the cognition, the behavior. And so this is what you're seeing here, and this is, again, not our work. What you're seeing here is imaging of the activity of a zebrafish brain as the zebrafish is thinking about whatever it is that zebrafish think about. But you can see this activity, and there's this hope called, of the field called neural decoding. The idea is that if we understood how to decode this, you could look at the electrical activity and figure out what is that animal, or in fact human patient, thinking about. What are the memories, the planning, the cognitive structure that is encoded by this. So it turns out what we've been able to find is that actually not only brains, but all cells in the body do this. So all cells have the same kinds of ion channels. They have the ability to pass this on to neighboring cells, and they form networks. And what you're seeing here is, for the first time, and this is a time-lapse video made by a colleague of mine, Danny Adams, who we use these voltage-sensitive dyes that basically show up as different colors based on the electrical activity of the cells. And so you're seeing the early few hours of a frog embryo, and all of these cells are communicating with each other to decide who's going to be head, who's going to be tail, left, right, belly, and back, and so on. And so very much like in the brain, all of the cells in your body, from the very first divisions after the egg is fertilized, start these electrical signals to figure out what they're going to do. And while these kinds of networks think about behavior, they think about controlling the body, these networks think about shape. They think about how to format and then repair the body. And so all of this works sort of like this, with very slowly changing voltage gradients, not spiking like you have in the nervous system, and nothing to do with, this is not at all having to do with like electromagnetic waves or holding your cell phone up to your head for too long. So these kind of, one of the first things we can do is we can detect these patterns. We can basically read the electrical memories of these cells as they're deciding what to do. And this is an embryo putting its face together, and you can see here that there's already in the electrical, if you just look at the electrical state of these cells, you can already see that it's planning to turn on the genes that will cause eye development, to do that here, and here's the mouth, and it's already laying out, it's like a subtle scaffold for the gene expression and the anatomy that's going to follow. But these are natural patterns, and you want to be able to track them and to repair them for birth defects and things like this. But there are also pathological ones. This, for example, is a tumor seen in an early tadpole, and you can see that already, even before the tumor shows up, you can see electrically that these cells have already severed their electrical connections to their neighbors. They're going to basically, they're about to decide that they are amoebas once again. They're going to treat the rest of the body as environment. They're going to move other places, that's metastasis, over-proliferate, and so on. So this kind of thing is sort of an obvious diagnostic, has diagnostic potential. Can we see this early, and can we intervene before the cells have really left the body plan? So in Planaria, what we've been able to do is to show that some of these patterns are, in fact, determinative of what happens. So when the head and the tail are removed, and you take this middle fragment that's going to regenerate a complete worm, there's this electrical pattern that tells you where the head and the tail are going to be. And so if we reproduce this head electrical state back here, what you end up with is a two-headed worm. And this is not Photoshop. These are real live animals that are in our lab. So you can make two-headed worms, you can make no-headed worms, and so on. And in fact, this body can contain a number of electrically encoded pattern memories for what it's going to produce if it is injured. And this was recently just on the cover of the Biophysical Journal, some of this work, and it was done by a graduate student, Fallon Durant. But one of the remarkable things about this pattern memory is that, like any good memory, once you've changed it, it keeps. So once we take a normal worm and we've made a two-headed worm out of it by changing briefly, 48 hours, that's all it takes, to control the electrical signaling, it makes this two-headed animal. From then on, forever, as many times as you want to remove the two heads in plain water, no more manipulation of any kind, this will now continue to regenerate as a two-headed animal. What we've done is permanently alter the electrical representation of what these cells think a normal planarian is supposed to look like. So once that pattern is changed, and it's stable, and it's a kind of memory, just like electrical memory in the brain, once you change this, the cells will continue to build to this pattern, although we can now wipe it back and go back and make them this way. So remember that this was all done without changing the genome. There's no genomic editing here. So the genome here is completely wild type. So if you were to sequence the genome of this guy, it would be identical to this. I mean, there's no difference. That is not where this information is. So think about how much there is left for us to do beyond just simply having a genomic sequence for these things. Now, I want to tell you a couple of stories for some of the undergrads that have made amazing progress in this field. So the first one is Maya Emmons-Bell. She was a Beckman Scholar in my group. She's currently doing a PhD in Berkeley. And she asked the following question. Well, this is nice and it makes 2 heads, but these heads are awful normal. What can we do to change this electrical activity? Might it be also involved in changing head shape? And what she discovered was there's a different way to perturb this electrical circuit to change the head shape to be like that of other species of worms. So here's a particular species of worm. You can see it's got a triangular head with a little auricles here. And when you cut off that head and let it regenerate while manipulating that electrical circuit, what it will do is it can make flat heads, like a P. felina. It can make round heads, like an S. mediterranea, or it can make its own normal triangular head. These species, oh, and by the way, it's not just the head shape, it's the shape of the brain and also the distribution of the stem cells are exactly like these other species. They basically get heads belonging to another species of animal. These guys are about 150 million years of evolutionary distance between this, and yet the genome is untouched. And so you can see here how important this bioelectrical sort of physiological layer of software is lying in between the hardware that's specified by the genome and the actual outcome. And of course, it's more than bioelectricity, so there are chemical gradients, there are physical forces, stresses, and tensions, and all of that. All of that is important. But it's simply amazing to what extent we can reprogram some of this software without having to change the genomic hardware. So she discovered this, and then after that, she found, so that was a nice paper in molecular sciences. But then she also discovered that, in fact, Planaria don't even have to be flat. So we were able to make worms that have this really strange spiky morphology, or this sort of cylindrical, it looks like a ski cap kind of thing, or even tubes coming out the back. So there are regions of the space of all possible Planarian shapes that evolution either has not explored or has explored and decided was not, fitness was not good, so you don't see them in the wild. But really, the range of anatomies that you can induce out of genetically normal Planarian cells is really extremely large. And so another student in the lab, and this is Jen Hamelman, who is currently doing a PhD at MIT. She studied both computer science and biology, and what she was able to do was to make models, brain-like models of cellular decision making. So she had a couple of nice papers as first author, as did Maya, showing that we can come up with representations very similar to what computer scientists use. They're called artificial neural networks, and they're sort of a basic paradigm by which people understand how information is stored and processed in computers and machine learning. So she was able to make these kind of closed loop systems where the network would assess and modify its own morphology. It would think about its own shape, and this is a pretty important advance. And one of the things that this kind of brain-like model suggested was, well, one thing that brains do is they represent the rest of the body. So in your brain, for example, in your sensory cortex, you have cells that map onto all the different parts of your body. So your hands, your fingers, the proportions of your face and so on are represented inside a region of your brain. And so another student, another undergraduate in the lab, Sarah, who's currently in training. She's an Olympic quality rower. She's currently, she graduated, she's practicing for the 2024 Olympics. She found the following remarkable thing, that it's not only the brain that represents distal tissues, but actually the rest of the body does too. So here's a froglet. It looks like this. It still has a tail, so it hasn't shed its tail yet. If this limb gets injured, so it gets amputated, and what the color here represents are, this is a bioelectric readout. We're able to see in the living animal the electric state, just like I showed you in the embryo. When this limb gets amputated right here, The opposite limb that was not touched in any way, it's uninjured, lights up in very much the same position. And so by watching the electrical activity of this limb within 30 seconds, it takes about 30 seconds for the information to get across, you can tell where the damage was and what kind of damage it was as well. So it's possible that lots of different body areas represent what's going on in other regions of the body, which is interesting not only from the basic perspective of how does information get around in the body, but you can think about things like surrogate site diagnostics. Can we look at a convenient portion of the patient to see what's going on somewhere else that might be difficult to image? And so she had a very nice first author paper in development. So this work is being pushed forward towards regenerative medicine. So here, frogs, unlike salamanders, do not regenerate their legs. So if you amputate a frog leg, you will either get a hypomorphic spike or nothing like that. And so what our team has done, and this is a postdoc, Celia, and then her two undergraduate students that worked with her, Christine and Haley, and this work is going towards developing cocktails of drugs which affect the electrical properties of cells to kickstart limb regeneration. So you see here we're starting to get toes, there's a toenail, eventually a pretty good leg, and you can see here when you touch it, the leg is both sensitive and motile, so it's actually functional. And so now, in collaboration with David Kaplan's lab, they're making these wearable bioreactors that are put onto the next model, which is a rodent model, and the idea is to really develop a supportive aqueous, almost embryonic-like environment with electrical, with what we call ionoceutical drugs, which basically control the electrical activity of the cells to convince them to rebuild the limb the same way they did during embryonic development. So this is an example of a regenerative medicine application. As well, we have another project which is worked on by other undergraduates where we have these human cancer cells, and we are attempting to basically reprogram them back to normal with a variety of electrical and light stimulation. So all of these things are really complex, and we are developing ways to try to infer novel interventions. So what kind of stimulus would give us a particular system, push it over to a healthy state from a disease state, and we're using techniques from machine learning and AI. And this was some advances made by another student, Taylor Malone, who he worked with one of our postdocs, Daniel Lobo, and their work actually ended up discovering a model of Planarian regeneration, which was the first model in this field discovered by a non-human intelligence. So what they did was to create a simple artificial cognitive system that was able to think about all of the different experiments that have been done in Planaria and come up with an understanding of what's going on. And to my knowledge, that's the first such that came from a non-human scientist. And Taylor was involved in that. So to close up, I just want to say there are lots of, this is just a small sample, there are many other undergraduates. We have probably on the order of 10 undergraduate students every semester working with us, and they do really remarkable work that pushes forward the frontiers of this interdisciplinary science. Most of them end up as co-authors on papers. Many of them are first authors on the papers. They really do incredible work here. And in fact, some of them start even earlier. So some students contact me even before they get to college. So this, for example, is Nina Ferenc, who got in touch as a beginning high school student, and she did work basically in her home and in her high school with me on the phone. And as a result, she got real data, and she ended up with a paper in macromolecular bioscience. So really, it's the drive and the energy of these students who want to tackle these difficult problems and really put in incredible effort that pays off. And I think Tufts is a pretty ideal environment in which they can do that. So I want to finish by thanking the various postdocs who have done some of the work that I've showed you, our collaborators. Of course, the animals that we work with, our funding support, you have to thank the animals, our funding support from the Allen Frontiers Group and several other foundations, and a special shout out for Susan Ernst, who's a member of the biology department with whom I worked when I was switching over from computer science to biology as a student here. And so this is 1992, that's my graduation from Tufts, and there she is, and she just retired very recently. But we worked together, and working in her lab made me think that it was possible to bring my skills from computers and engineering into biology and really do something relevant. So I will close there, and just to show you that if you're wondering what two-headed planaria look like when they're moving around, this is it. So thank you, and I'll be there for questions. I'll be in the back.
So as everybody is fascinated by that as I was, I never stop learning something new every time. So thank you so much, Professor Levin. That was fascinating. We have a full day for you planned. We have a number of sessions going on next. In this room will be Laura G. talking about economics, and check your brochures, and there should be a slide coming up that will direct you in a moment to the next locations. Have a wonderful rest of the day, and thank you for coming.